From 2016 to 2018, a large science and industry collaborative study was undertaken by Biosecurity New Zealand. It was designed to better understand the effects of disease, climate and apiary management on the health and productivity of New Zealand's honeybees.
The Bee Pathogen Programme is a 3-year research project that is based on international studies where we look at honeybee health across the country. What do you think of that? Starting up, eh?
Yeah.
There was 60 apiaries nationwide, stretching all the way from Cape Reinga down to Southland and some of the offshore islands as well. And we go to those apiaries that we identified and interview the participant beekeepers about work practices they had, their productivity in their colonies, their various management practices. We would also take samples of bees. Those come back to our laboratories in Wellington and Auckland, and we test those for a wider range of diseases and microorganisms, bacteria, viruses, parasites. And we're looking for things that are known to circulate within New Zealand. We would call those endemic organisms. And we're also checking to make sure we don't have exotic organisms, things that have come from outside of New Zealand. We know Varroa is still present in New Zealand. It arrived year 2000. That is still a threat to beekeeping on a daily basis. American foul brood is a disease, something that beekeepers have to manage as well. And then we have some organisms that we don't have in New Zealand. And those are things, of course, we want to keep out, and that's part of our role here. Those things like European foul brood that we don't have here, we're taking an interest in making sure those aren't present. Small hive beetles, another one that we keep a close eye on. One of the central reasons for us doing this work is to say, well, here's more evidence that we know we don't have these exotic organisms, but also for the things that we do have, this endemic disease, these things that are known to circulate in New Zealand and some of the things that we really would like to get rid of, what kind of practical advice can we give to beekeepers from the scientific work that enables them to meet that goal as well? That is the goal of the Bee Pathogen Programme.
Bees form an integral role in New Zealand, and not only in their export value and the production value that they produce. I think they bring in over $350 million for New Zealand, but just the role they play in pollinating and supporting other horticultural industries in the country. So we have over 1 million registered beehives in the country and 8,500 registered beekeepers in the country, and so they all form a significant component to our primary industry and the contribution that the beekeepers and the bees play for New Zealand. We had a wide array of people involved, which we're very grateful for their involvement. The volunteer beekeepers who let us come back every 6 months and sample their hives, trained apiary inspectors that went to the hives, carried out the surveys and sampled the hives for us. Not only that, we had a lot of support from industry itself and the industry bodies that are there. And we've also had a lot of support from MPI to carry out this research. It was a collaboration between our two labs, so the Plant Health and Environment Lab and the Animal Health Lab based here in Wallaceville. And the project itself was supported by the Operational Research Grant, which helps us become more develop capability in a lot of things that MPI does and put it into everyday use. Follow on from the Bee Pathogen Programme is the AP Wellbeing Project. We've spent 2.5 years carrying out the Bee Pathogen Programme, and now the AP Wellbeing Project is to produce management guidelines and usable resources for beekeepers to put all of this wealth of scientific data that we have into everyday best practice resources for beekeepers to improve their management.
When we receive the bee samples from the beekeepers at the Wallaceville laboratory, we do DNA, RNA testing. So even bacteria and viruses have DNA, and through that we can apply very sensitive tests and very quick, efficient tests to see if those viruses or bacteria or even parasites like Nosema are present, and we can do that with high precision. And for the participants in the Bee Pathogen Programme, we go back to those participants, constantly updating them on how their sample was tracking and how it was tracking against other areas of the country with the very special precaution that we keep all the information anonymous. The confidentiality of our participants is something we greatly treasured. And at the end of the 2.5 years of collecting the samples, we can then go through, take all the information about how many viruses, how many bacteria, how many parasites were in each sample, and collate that with all the other information we gathered from the beekeepers through interviews. So productivity, colony losses, how they treated for varroa. We are now starting to look at how those factors affected disease levels. Results from the Pathogen Programme has replicated some previous academic studies that were done, as we understand that between 40 to 60% of apiaries have varroa at detectable levels in it, not necessarily causing a problem, but we do know it is around New Zealand. We do see that Nosema can be common in apiaries, but not necessarily causing a problem, and that is what we want to look at from our data, is the factors that make that Nosema become a problem. So we also see viruses that we know are already in New Zealand. We see those quite common amongst colonies, but the interesting thing for us that we're undergoing the analysis is whether those viruses, when they are present, cause a problem or don't cause a problem. So it's going to be a combination of factors, and it's those factors that we are excited to be able to talk about in the coming year as we're doing the analysis.
Thanks, New Zealand On Air.